Today I will talk about a new approach or new methods to study the effect of the radiation on biological molecules. So we try to mimic the nature to have much more simple systems to study the nature. So the motivation for this work can be these works about the effect of UV in DNA. This now is well known. UV creates damage the DNA, but if we study the system, the DNA in vacuum, the effect of the degradation of the bases, which is related with the 260 nanometers peak, this peak here, practically the bases are not damaged with UV. But we put the UV, the DNA in water. It is a aqueous solution of DNA and irradiated, in this case with the 254 nanometers. We can observe a decrease of the band associated to the DNA base, and this is decrease of this band. So, for degrade the DNA, it is need the presence of the water. So, if we study the DNA molecule or biological molecules in vacuum or without water, we have other processes which are not involved in the nature when DNA is in the cell, which is in solution. So we can systemize the hypothesis to study the effect of radiation on biological molecules as in vivo, in cells or in the humans, or study the molecules in solution. Here we can use several techniques to study the degradation, or study individual molecules in vacuum. But here, in this case, we have some difficulties because we can study only small molecules. If you want to study proteins or we need to study individually on a surface, and in this case we can use the techniques of characterization of surface. So the other hypothesis, or finally, we can study in thin films and prepare films of biological molecules with lipids, DNA, proteins, what we want. So, but for prepare these films, we need to put water inside of these films. If you see the cell, we can see the cell is composed by the membrane, by a lipidic bilayer, which can easily represent here. So the idea is prepare liposomes, a system prepared with lipids, and put inside the molecules that we can, or molecules that we can study. So we have a much easier system to study, and now put these in films and use all the techniques of characterization of materials. To prepare this lipidic bilayer, we can use the Langmuir-Blodgett system, but we need to take one layer, and after that the second layer, and it is possible also to include some molecules, and we have the proteins inside of lipids, but we need to take off of the water this membrane when we put in the tube of the Langmuir tube the support and take off. So the other hypothesis to prepare these films are the layer by layer technique, which is a very simple technique and allows to prepare by layers, followed by layers of several systems. For this technique, to prepare one type of a film, we need to have solution of a polyelectrolyte solution or a cationic solution, and start to absorbing on the support the molecules in the polycation solution. This is the example, and after that take off of this solution, wash to clean all the molecules which are not adsorbed, and now put the support with one bilayer in the polyanionic solution and wait some time to absorb, wash, and we have one bilayer. It is a very simple technique and allows to prepare different systems. As we are interested to prepare biomimetic membranes and rudimentary cells as the liposomes, as the case of liposomes, as the model for these rudimentary cells, we start to a very simple system to prepare films with lipids. And in this case, we use a polyelectrolyte, cationic polyelectrolyte, pH, and this lipid, DPPG. We prepare these films, layer by layer films, and measure the ultraviolet spectra of these films, and measuring the maxima of the absorbance peak, we can see that these films grows linearly with the number of bilayers. But it is interesting, after characterizing the adsorbed amount in these films from the UV spectrum, we can obtain values of 6 milligram per square meter, which is related with one bilayer. So, when we prepare these films, we have bilayers adsorbed between polyelectrolyte layers, which is also in accordance. The values are in accordance with the Langmuir films on surface and on the Langmuir tube, which has the value of 2.1 10 18, which is very close of our value obtained molecules per area is the double of the values in the Langmuir tube, which indicates that we have a lipidic bilayer. Also, the ruggedness of these films are very low. These films are flat. So, this system, we can use this as a biomimetic membrane. Meanwhile, we study the effect, we try to encapsulate molecules inside of liposomes and study another system, melanin is a biopolymer which is in our skin and has some properties, fluorescent properties, photoconductivity and piezoelectricity. Several can be used in several applications. This is the molecule of melanin, and we try to prepare films with the pH, and we cannot prepare these films. And after that, we try with another polyelectrolyte, this polyethylene imide, PAI, and also we have difficulties to prepare films of the melanin and this polyelectrolyte. So we try to encapsulate melanin in DPPG and prepare films with that. In this case, we observe that these films grows and grows practically linearly, and after, in this case, 20 days, these films present emission spectra. So melanin has its properties, and also we can observe fluorescence decay in these films. So melanin, which lose all its properties in the case of the polyelectrolytes, only between the polyelectrolytes. In presence of DPPG, we can have the properties of the melanin. Why this and the values associated with these films reveals the thickness reveals that the liposomes are integral after adsorption. Why we observe these liposomes integral in the surface? In the first case, we cannot do that. The reason for that is the PAI is a polymer, is a polyelectrolyte, but is a polymer which after adsorption gives rose surface. So these results lead us to study the effect of the roughness on the adsorption of the liposomes, DPPG liposomes. In this work, we start with one layer of pH, which is a very flat surface, and we absorb in this case, one lipid bilayer of DPPG. And this is the kinetics, it's the absorbed as a function of the time. So the liposomes adsorb and open in the surface, and you have a lipidic bilayer. But as we increase the ruggedness given by this cushion, initial cushion of polyelectrolytes, the lipids are being adsorbed in the totality, and after that we start to obtain films with liposomes. The values associated to this adsorbed amount corresponds to a whole liposome, and these values are associated to a lipidic bilayer. This is the values of ruggedness associated during the adsorption, this is interesting, but I think it's not necessary to enter in these details. But this small figure can show that when we have the ruggedness, the curve of the ruggedness, the fraction of open liposomes decrease. So after 5, in this case 6 nanometers of the ruggedness leads to adsorption of liposomes with about 15 nanometers of diameter. So now we know as prepared lipid bilayers and liposomes in surface. These films have several applications. It's not only for the case of radiation, but here I will bring you an example where the effect of radiation can give us several informations. You use DNA film, DNA film, and measure the conductivity of this film with the frequency, and irradiate the film with the time. This is the curves of the conductivity, the real conductivity of the sample, and taking the data obtained at 1 megahertz, this data here, we can study the conductivity using the correlated barrier opening model. And when we use this model to our data, we calculated a hopping distance of 3.4 angstroms associated to the hopping of the electron on the DNA molecule. This value is quite interesting because it's the distance between the base pairs and phosphate groups in the DNA chain. So we take our data in the effect of UV on DNA molecules measured by infrared, and we have this in the case of phosphate groups is this, and we plot the conductivity of the sample, which is associated concentration of pair sites, is this square here in the graph, and the squares are associated to the infrared data, and this is the red points. The red points are the concentration of pair sites and the number of phosphate groups on the sample. And we can observe the similar decrease. So we conclude with this work that the DNA can be used as UV sensor, but also we measure, we associate the conductivity of the DNA to electron hopping in the chain. The hopping distance is 3.4 associated to base pairs and phosphate groups, and this is for the first time measured. In the literature, the DNA is quite difficult molecule to study the conductivity, because someone says that DNA is conductive is semiconductor or is insulator in certain cases. And this is given by the number of phosphates which are in the molecule. If the molecule was in presence of radiation, UV radiation, it loses the phosphates and it loses the conductivity. This work, after we obtained this work, we see in the literature only 3 works, theoretical works, which speaks in the opening, in the electron opening in DNA molecule. Other application of these films, it is in the sensor of determined molecules. This is, I bring you the delta neutrino sensor, which is a vineyard pesticide. And we can have sensitivities about 2 concentration 10 minus 11 molar. Is very interesting when we try to find very small amounts in a solution. So, if we have a problem, we can prepare systems for a study or for prepare sensors or other devices. So, we can absorb lipidic bilayers. If we have flat surface, or if we have rugged surface, we can absorb the whole liposomes. We can prepare in the beginning solution or more emulsion with the other molecules, and we can have lipidic bilayers with some proteins or the other molecules. We can put here molecules inside or also in the wall of the liposomes, and in this case it is quite interesting for the case of sensors that we can put here proteins or some other molecules which have sensitivity to a determined molecule. So, thank you very much. I think it's not necessary to repeat my conclusions. So we have some questions.
On those models, where you have a membrane immobilized on the field, the B model, was this model ever used for activity due to the reaction of an enzyme, for instance? Because it should be very interesting.
This can be used, yes.
Is there any study?
I think you have some studies in that.
With the enzyme activities?
Yes, in the case of sensors, I know in the case of sensors, they put some molecules and prepare, in this case, lipidic bilayers.
Without the case of sensors, for instance, to study a particular protein as well.
Yes.
You are showing these for protocols, which are the best?
Depends on the situation. If you want to study a lipidic membrane, you can use this, or to transport in the membrane, or in the case of this in the case of Ciências, can use this system. If you want to study the molecule inside of liposome to study the effect of radiation, we can use this with the molecule inside. It should be interesting to study the DNA inside of liposome to study the effect of radiation. Or some, in the case of your proteins, to study the proteins inside of liposome. In this case is much interesting for the case of sensors, where we have here some molecules which can detect the molecules in a solution.
Okay, so we'll thank the speakers again.
Good morning. I will be speaking about oxidative stress sensors, as Pedro said. And first of all, I know that a lot of people have already talked about oxidative stress, or ROS, reactive oxygen species. But I had thought of doing a little bit of introduction about how the oxidative stress affects our organisms, us as humans, and also bacteria, unicellular organisms. So, oxidative stress may come from smoking, alcohol, the diet, medications, air and water pollutants, fast foods, and stress. Even our stress causes oxidative stress. Well, just about everything we do results in oxidation and production of potentially damaged free radicals, or not only free radicals, but the reactive oxygen species. Reactive oxygen species may be radicals or non-radicals, as you know, and the one I'll be most interested about will be hydrogen peroxide. The formation of free radicals, as you know, may be because of UV light, ionizing radiation. Smoking also may form hydroxyl because of first forming superoxides, or even NO, which is not really a reactive oxygen species, but more a reactive nitrogen species, but which is also very important in oxidative processes and also in cell signaling in higher organisms. Also air pollution, which also produces hydroxyl. Cellular metabolism produces a lot of hydrogen peroxide and hydroxyl, and our immune system, as a way of combating infections, produces also a lot of ROS and even NO also. So all of this results, as many people have already said, in DNA damage, but not only in DNA damage. Well, here I'm not going into this, but hydroxyl is the major species that causes DNA damage, as you know. But not only DNA damage, but also protein damage and lipid peroxidation, which are also major factors in affecting our organism and producing diseases. So I have here an example of Parkinson's disease. and also to correlate it with the conference that Elise gave yesterday about ferritin, and also because I'm also involved in the ferritin work, I decided to show a little bit of how hydrogen peroxide and iron can lead to Parkinson's disease. As a result of aging, ferritin, the enzyme that Elise described yesterday, decreases in concentration as a result of age. Also, the blood-brain barrier becomes leaky, and all of this produce a higher free iron concentration. And this, when it encounters hydrogen peroxide, produces hydroxyl. And hydroxyl, as you know, will cause lipid peroxidation, protein misfolding. Protein misfolding causes aggregation, and then finally it leads to neurodegeneration and cell death. Okay. So some of the diseases that are linked to reactive oxygen species. Diseases of the respiratory system are mainly due to inhalation of oxidants. In brain, we have Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis, Down syndrome. We have diseases that are related to cardiovascular system, like heart failure, skin diseases like porphyrias, which are also related to oxidative stress, muscle diseases, muscle dystrophy, and nowadays we also think that aging is a result mainly of damage due to reactive oxygen species. Cancer is also related, diabetes, and even autoimmune diseases are also related to the building up of reactive oxygen species. Well, in the organism, reactive oxygen species may be formed due to, we use oxygen as a final electron acceptor, and in mitochondria, this process is not always very, how do I say, very accurate. So sometimes other species are produced. When we reduce oxygen with only 2 electrons, hydrogen peroxide is produced. So we have to have enzymes that deal with these species that are produced. Otherwise, we wouldn't be able to use oxygen as a final electron acceptor. So in our organisms, we have superoxide dismutase, which uses superoxide and turns it into oxygen. We have catalases, which use hydrogen peroxide and turn it into oxygen and water. And we have peroxidases, which reduce hydrogen peroxide to water. But also, the existence of hydrogen peroxide may lead to the production of other species, of other reactive oxygen species in the organism. Like if you have chlorine and hydrogen peroxide, we can produce hypochlorite. If you have superoxide and NO, you will produce peroxynitrite, and this is very reactive also. And hydrogen peroxide, if it meets iron 2, by the Fenton reaction produces hydroxyl radical. And hydroxyl radical is one of the main oxidative species in the organism. But reactive oxygen species are also important because they are not only bad for the cells. Cell-specific cell signaling pathways will not work, and you have cell death, diseases, aging. But if you have less ROS than you need, also your immune system does not work, and other also signaling pathways where NO is involved will not work. So the organisms need to have sensors, proteins that sense the amount of ROS and that allow for it to be balanced and always more or less in the same level. So these sensors may act in two different ways. They may be activators or repressors of the transcription, or they may act and activate protein kinase cascades, and which then activates transcription factors. This will result in gene transcription. And which kind of genes? Well, what I'm going to talk about today is about a peroxide regulator repressor, PER, which is a bacterial hydrogen peroxide sensor. It regulates the expression of ROS defense enzymes in bacteria, and it has two metal centers per monomer. One is the regulatory center, which can bind iron or manganese, but the difference is that when you have manganese in this site, the sensor is mostly insensitive to hydrogen peroxide, which means that when you have manganese, the sensor does not react to hydrogen peroxide and does not become inactivated and like I'm going to describe. And we have a structural center which binds zinc. The importance of these repressors is mostly to pathogenic bacteria because, as I told before, immune system generates high amounts of ROS. So for pathogenic bacteria, this will be essential, an essential repressor. Okay, so the regulin, which are the enzymes that are regulated by this sensor, includes catalases, peroxidases, DPS, the protein that Elise talked about yesterday that uses hydrogen peroxide and iron to produce rust inside, to accumulate iron inside and as a storage. And also in some bacteria includes superoxide dismutase, includes in biosynthesis enzymes, and also the iron uptake regulator. The iron uptake regulator is the protein which also regulates all the iron, the proteins necessary for the uptake of iron. So they regulate, FUR regulates PER and PER regulates FUR. Okay, so the protein which was first discovered, this protein was first discovered in Bacillus subtilis. And this is the structure of the Bacillus subtilis PER. And we have an N-terminal DNA binding domain. And in the C-terminal, we have the dimerization. This is one monomer and another monomer. Each monomer has an iron iron or manganese binding site and the zinc binding site included in the dimerization domain. The regulatory center, which can bind either manganese or iron, is bound by 5 amino acids of the polypeptide chain, 2 aspartates and 3 histidines. From the mechanism that is published, the key amino acid would be this histidine 37, which is oxidated when the protein encounters hydrogen peroxide. But recent results have revealed that this aspartate also is essential, although it is not oxidated, although we do not know what happens to this aspartate when the protein meets hydrogen peroxide. But without this aspartate, per becomes like a full protein, becomes responsive to iron, the presence of iron, but does not respond anymore to the presence of hydrogen peroxide. So, the protein has 2 forms. An open form, when it does not have iron or manganese, and when it binds iron or manganese, it has a closed form, and this is the only form that is able to bind DNA. The hydrogen sensing mechanism which is described would be something like this. In this mechanism, the only role of iron would be that of producing hydroxyl radical, and this hydroxyl radical would in turn oxidize one histidine, the histidine 37, and with that histidine oxidized, iron comes out of the protein, and we have again this open form, but now with one histidine oxidized, which does not allow for iron to enter again, and the protein would be used then would be marked for degradation. So the mechanism would be something like this. We have the open form with zinc, a dimer, a protein dimer, and when we add iron or manganese, the protein changes its conformation and becomes in what we call the closed form, which is able to bind DNA, and when the protein is bound to DNA, there is no transcription, so it is a transcription repressor. When hydrogen peroxide is present, the histidine would become oxidized, the protein would adopt again the open conformation, and iron would be lost, and now protein is not able to bind DNA, and transcription would occur of the defense enzymes. So in the presence of hydrogen peroxide, the protein does not repress transcription, and the enzymes like catalase or DPS would be produced. So, sorry. What we devised when we started working with this enzyme, what we had to devise first was an activity assay to understand in which conditions the protein is active and inactive, and to be able to then interact it with hydrogen peroxide and other oxidants as we intended to do, and understand in which conditions then we get the protein in an inactive form. So what we started to do was to clone, take a fragment of the DNA where the protein binds, which is called the per box. It's a sequence of DNA where the protein binds, and then when we add the protein with zinc and iron, it would bind, and when we add the protein, for instance, without iron, it won't bind. If we cut with a restriction enzyme that cuts precisely in this sequence, in the per box, what we get at the end would be if the protein is bound to DNA, we have the DNA is protected, and at the end we have the same fragment. And if the protein is not bound, then at the end we have the DNA hydrolyzed. What we in fact did was to clone this sequence in a plasmid, and then try the activity assay when we add the restriction enzyme. If we have a plasmid, at the end we have the linear plasmid, and if the protein is not bound, at the end we have, here we have the circular plasmid, and at the end, if the enzyme is not bound, you have the linear plasmid. But what in fact happens is that this complex, when we run this DNA in electrophoresis, gel electrophoresis, what happens is that this complex stays in the well, does not migrate in the gel, and when we have this linear DNA without the protein bound, then we have a band which corresponds to the linear DNA. So what we realized was that when we added, we had to clone the protein. We have to find a way to produce it without any iron, because if the protein sees iron throughout the purification process, at the end we have the protein with the oxidized histidines, and which is not active. And when we finally did this process of purifying the protein without iron, at the end we had the need to understand if the protein was active or not. So we added zinc and iron, and we realized that the protein was active because it formed the complex with DNA, and when we added hydrogen peroxide, the complex is still a little bit of complex in the well, but you can see the linear plasmids cut with the restriction enzyme, which means that when we add hydrogen peroxide, the enzyme does not bind to DNA. So then we did mass power spectroscopy to understand how the protein, if the protein was in an active form, and when we added iron and zinc, if the conformation changed or not. And when we had a preparation which only had zinc, and one preparation which had zinc and iron. And you can see from, well, I have these lines here that correspond to the peaks in this spectra of the closed form, and you can see that the peaks are not the same in the open form. And I did the different spectra between the open and closed, and you can see that we have different species here. And any of these species do not correspond to iron being solution. So iron actually enters the protein, even though we produced it without iron. Iron actually enters the protein because none of these correspond to iron in solution. So what the questions that we want to answer now would be to understand the iron role in the sensing mechanism, because all the studies that have been done so far were done with the manganese form of the protein. No one has looked into the iron in the protein, because it is very difficult to manipulate the protein with iron. It's very sensitive to oxygen and hydrogen peroxide. And even when we are purifying it, as I said before, if you have iron in the protein, at the end you get inactive protein. So we want to understand the iron role. How do we plan to do it? We plan to do kinetic studies and use mass power spectroscopy as a primary tool, because it sees all the iron states. And we want to do kinetic studies, freezing the samples in different times to understand if we have different intermediates developing. And also, we want to understand why is aspartate 104 essential for hydrogen peroxide sensing. For that, we plan to do mutants. And more important than this, what I was planning to do was also to understand if per is also a sensor of other reactive oxygen species, like and also reactive nitrogen species like NO or superoxide, and try to do also the interaction of the protein, the loaded protein, with all of these species, and understand if it also reacts to these species. And finally, understand how per responds to ionizing radiation. If you expose the cell to ionizing radiation, we all know that a lot of things happens, but how do the sensors, how do the oxidative stress sensors behave under ionizing radiation? That is something that we also want to understand. Finally, I would like to acknowledge the people that have worked in this work, mainly Vasco, which is my master's student, and Alice and Pedro, which are always present in all of this work. Thank you.